Good morning. I am Vikas Tudeja at University of Alabama at Birmingham. We'll get started with the next session to be on time. Actually, one of the talk is canceled, the second one, so we will have some more time for discussion and question and answer session. So this session is tumor initiation, progression, and metabolic stress in PDAC. The first talk is by Hannah Allele, Metabolism, Autophagy and Cachexia, and the Immune System. Dr. Allele is from Comprehensive Cancer Center, Munich, Technical University of Munich. Welcome.
Thank you. This one? Oh, yeah, okay. So good morning, everybody. Thank you, Vikas, for the introduction, and thank you for having me here. Indeed, I felt obliged to come all the way from continental Europe, Germany, to Miami to visit the APA to meet all the icons of pancreatology. Again, I think personally, I felt that this is important in these days. Also grateful to be given the opportunity to share with you our perspective on how metabolism interacts with the immune system in pancreatic cancer, and also giving a glimpse on what is currently done in the scientific field. But given the time frame, I will not be able to go into very much detail, so just giving spotlights on the field. So, okay. So when we talk about pancreatic cancer, I think one important issue that we should bear in mind is the morphology of pancreatic cancer. On the right-hand side, you can see a classical histological feature of pancreatic cancer with a strong desmoplastic stroma, and in between the tumor cells. And this picture very much resembles a desert-like situation where cancer cells are more or less the oasis, being surrounded by a hostile environment. And looking at this picture, the question is, how can cancer cells survive in this hostile environment is an imminent question. So in fact, the issue of how can cancer cells survive in this environment is something that is not entirely new, because it was Otto von Warburg who was the first scientist who addressed the issue, how do cancer cells metabolize and how do they utilize glucose? And if you look at this grant proposal in the 20s of the last century, it was only one sentence asking for 10,000 German Reichsmark, which equals a medium-sized grant nowadays. That was not a very much elaborated proposal, but he delivered. He delivered many issues to the field on how glucose is metabolized in cancer cells. And in fact, he was honored with the Nobel Prize in 1931. So that was the first insight in how cancer cells can utilize nutrients other than normal cells. Somehow his contributions were taken away, or the importance of his contributions were taken away by the discovery of the DNA and also by tumor suppressors, tumor promoters. So by this, the metabolism of cancer cells was not that much in the focus of basic scientists. So somehow he got not forgotten, but he was not that important as proposed by the DNA guys who identified tumor suppressors and tumor promoters. Nowadays, by focusing on the genetics of pancreatic cancer, we have done a huge work in identifying the genetic landscape of pancreatic cancer. Nowadays, in the era of personalized oncology, we are trying to find the needle in the haystack or hay ball to find genetic vulnerabilities in pancreatic cancer to treat those patients. But at the same time, we have realized that there is a link between genetic mutations and metabolism, identifying works done by Ron DePinho showing that KRAS mutations induce a rewiring of metabolic pathways utilizing specific nutrients in cancer cells, specifically in pancreatic cancer cells. So somehow there was the link that genetic alterations can also induce metabolic pathways to utilize nutrients in cancer cells. So that was the point where metabolism and genetics met each other again. So at the end, just focusing on metabolism, we have realized over the last few years by works done by many groups all over the world that metabolism is very important in pancreatic cancer specifically, but generally in cancer as well. And for pancreatic cancer, we have learned that metabolism is very important for many features that are characteristic for pancreatic cancer, the so-called hallmarks of pancreatic cancer. But how do you want to address metabolic pathways in pancreatic cancer, and how do you want to elucidate all the contributions of metabolic pathways in the conjunction of genetic alterations? If you look at the metabolic pathways that are important, you will see that it is a huge work to be done to identify the contributions of metabolic pathways to cancer cell survival. So it's looking like a metro map, and you do not know where to embark and where to get out. So it's very sophisticated to target specific metabolic pathways. But anyhow, we have to address this issue. And in the last 10 years, metabolic pathways were focused on. And the most important one that was elucidated and elaborated by Alec Kimmelman's group is the process of autophagy. Autophagy is a multi-step or multi-step process in which organelles are recycled to provide cancer cells different nutrients. So for checkup of the function of organelles. So it's a very conservative pathway that is important not only for organelle recycling and degradation of multimeric proteins, but also providing cells nutrients. And in fact, this process of autophagy has been shown to be highly activated in cancer cells in vivo as well as in vitro. And the most important inhibitor of this pathway of autophagy has been hydrochloroquine. And in the study of Alec Kimmelman on the right-hand side of the slide, you can appreciate that treating xenografts with hydrochloroquine was sufficient enough to induce tumor regression, or at least to inhibit tumor growth. So there is evidence that metabolic pathways regulated potentially by autophagy are important for the survival of cancer cells and could be an interesting therapeutic target. But if you go to the genetics of this pathway and you delete ATG5, which is a very important protein in the process of autophagy, you will see that you will have a significant impact on pancreatic cancer development. However, the impact are very controversial. There were two studies deleting ATG5 in the pancreatic xenograft autochthonous model, and you can see on the slide that the conclusions were not really conclusive, with studies suggesting that deletion of this ATG5 acts as a tumor suppressor. Other studies suggested that ATG5 deletion induces tumor, so it's a tumor promoter. So what is the reality? So we would expect that this process is very sophisticated. They will not come to a very clear conclusion at the beginning. But what we did when we started to look at the data, at the same time we did also this study, but they were faster than we could see that here the heterozygous deleted mice, they had a survival that is between the homozygously deleted and the wild type animals. So what is the importance of the genetic deletion of one allele of ATG5 in pancreatic cancer? And that was the reason why we took over our studies and gave them another focus, focusing on heterozygosity of ATG5 in the pancreatic cancer model. And in fact, by doing so, we had a very interesting observation. Beside the fact that we could demonstrate that autophagy is a process that is activated through the entire panin progression, we have seen that allelic loss of ATG5 in this mouse model gave rise to more tumors, but most strikingly to more metastatic tumors. So that was very surprising. That was somehow an observation that was in between the two classical studies showing the genetic deletion on both alleles suggesting either tumor promoter or tumor suppressor. It seems that the heterozygous deletion of ATG5 is somehow impacting metastatic load in this mouse model. So we went on to elaborate in more details this observation. And what we did is we isolated tumor cells to see whether this effect is a cell autonomous mechanism. And in fact, by doing so and isolating tumor cells, we could see that the proliferation of these cells were not different. However, if you put them on matrix, on a Boyden chamber assay, you can see that they are more invasive and more migratory cells than the wild type tumor cells, suggesting that mono or heterozygously deleted ATG5 in these tumor cells influences metastatic capacities, but did not have any impact on proliferation of the tumor cells whatsoever. So we elaborated in more details to be sure that this cell effect is cell intrinsic, and we did many other experiments showing that IV injection of isolated tumor cells in fact increased metastatic load, and we did siRNA approach to show that dosage dependent down regulation of ATG5 impacts metastatic load. So it seems that this is a cell autonomous effect. To work more details on this, we did some supernatant analysis of these ATG5 heterozygously deleted cancer cells, and we observed that these cells secrete into the media many cytokines and chemokines shown here, but also metabolites that in total polarize macrophages towards M2 macrophages, suggesting that the heterozygously deleted tumor cells does not have only cell autonomous effects, but it has also an impact on the microenvironment through the secretion of chemokines, cytokines, and metabolites, thereby impacting macrophage polarization. And in fact, we did also some in vitro experiments to show that this effect is indeed true, even in IV injection transplantation models and co-incubation of ATG5 heterozygously deleted cells. The supernatant of these cells induced raw macrophages towards M2 polarization, suggesting that in fact the tumor cells that lack one ATG5 allele can also impact macrophage polarization. And the metastatic phenotype seems to depend on the macrophages. By using clodronate, we could show that this is indeed the effect of macrophage polarization in the in vivo setting. Now, this is another observation that we are currently working on, and it's not published. It is currently in preparation, showing that we do not have only cell autonomous effect in ATG5 heterozygously deleted cells. We did not only have microenvironment issues, but seems also macroenvironment, because injection of these tumor cells into xenograft models induced the upregulation of FABP4 in hepatocytes, suggesting that there is something secreted by the tumor cells that impacts the activation of hepatocytes, potentially influencing pre-metastatic niche. So this is an observation that we are currently working on and try to elucidate in more details what is the impact of ATG5 in the tumor cells and the hepatocyte activation of FABP4. So this is just showing that what we have done is somehow human relevant, because we never see ATG5 loss in human pancreatic cancer. What we see is different levels of ATG5 expression in human pancreatic cancer, suggesting that we have to stratify in more detail these patients for therapeutic studies according not to the deletion of ATG5 homozygously, potentially by stratifying the patients according to ATG5 expression levels, where we could see that in fact more ATG5 is associated with less metastatic load in patients with metastatic pancreatic cancer. So this is something that we are pursuing in more detail. On the right-hand side, you can see the study that were already done in ATG5 and pancreatic development and pancreatic cancer. So it's more complicated basically as we expected. But coming back to the human situation and to the title of the talk, what is the impact of metabolism of immune system? I think I could show you that ATG5 has an impact on the microenvironment through M2 polarization. And there were also two other important studies suggesting that interfering with autophagy has an impact on the activation of the immune system in the microenvironment. And I think these are very important observations because they could guide us through more designed clinical trials. Because if you look at the clinical trials that were started as soon as after autophagy has been identified as an important target in pancreatic cancer, clinical trials were started with hydrochloroquine in combination with gemcitabine and Abraxane. And as shown from our basic study experiment, in fact, these patients did not better. They even had a worse prognosis under the combination of gemcitabine, Abraxane, and hydrochloroquine, suggesting that there is something to ATG5 level and the metastatic load in human situation as well. So I think in the future, we have to guide ourselves more detail through clinical trials with interference of autophagy inhibitors, and we need to identify better stratifier than we did in the past with the clinical trial, which failed here, just showing as an example for many other studies that failed. So let me conclude. So autophagy is still an interesting target, but I think we have to give more input into stratifying the patients according to autophagy activation and autophagy inhibitors. And the most important observation nowadays that were reported by many other groups and also reported by our team is that inhibition of autophagy influences the activation of the immune system, and that could open up a new field in the combination of autophagy inhibitors and checkpoint inhibitors, something that we should definitely, seen from the clinical point of view, follow up in more details. And I think that we can get more out of these observations than we did with the pure inhibition of autophagy in combination with chemotherapeutics in pancreatic cancer. So with this, I would like to thank you for the attention, and I think we'll take the questions later, right?
Yes, we'll take the questions in the end. Next talk is by Dr. Rosalie Sears, who is Professor of Molecular and Medical Genetics and co-director of Brendan Colson Center for Pancreatic Cancer, School of Medicine. The title of the talk is Cmic Signaling in Pancreatic Cancer.
Hello, I'd like to thank the organizers for the opportunity to present our work on Cmic in pancreatic ductal adenocarcinoma. I have the following financial relationships to disclose, and I will not discuss off-label drug use. As I'm sure most of you know, Cmic is a transcription factor that binds to EBOX sequences in DNA along with its partner protein MAX, and this facilitates the recruitment of transcriptional coactivators that lead to the release of polymerase and transcriptional elongation. Genomic studies have demonstrated that Cmic is bound at all actively transcribed genes and functions as an amplifier to increase their expression. At oncogenic levels of expression, Cmic can invade into more closed chromatin to initiate new transcription. Together, this allows Cmic to control all of the hallmarks of cancer, including cell growth and division, blocking differentiation, increasing cell motility, and increasing cellular metabolism. In addition to these cell intrinsic activities, activation of Cmic in tumor cells leads to microenvironmental changes, including increased angiogenesis and the induction of an immune suppressive tumor microenvironment. The power of Cmic in pancreatic ductal adenocarcinoma was recently shown by an elegant mouse model in Gerard Evans' lab. This schematic is taken from a review written by a talented graduate student in my lab, Isabel English. The authors of this study demonstrated that expression of KRAS in the acinar cells leads to the induction of pancreatic intraepithelial neoplasias, but does not progress onto PDAC unless you activate Cmic. In this mouse model, they have a switchable Cmic such that they can turn on Cmic with tamoxifen, and in that case, the pannins progress to invasive PDAC. Along with this, the tumor microenvironment expands into a desmoplastic immune suppressive microenvironment. In order to mimic therapeutics, this switchable model allows the authors to turn Cmic back off again. And what they observed is that the desmoplastic immune suppressive tumor microenvironment collapses and anti-tumor immunity is induced, and the PDAC is resolved into pannins all the way back to normal acinar structures. My lab has been studying post-translational regulation of Cmic activity for many years. We've demonstrated that extracellular signals that activate new Cmic transcription, as well as downstream kinase cascades, including the RAS-RAF-MEK-ERK kinase cascade, leads to the phosphorylation of Cmic at a conserved site, serine 62. And this phosphorylation event increases Cmic protein stability. It also makes for a docking site for the PIN1 phosphorylation-directed PIN1 proline isomerase that induces a transcis isomer change of proline 63. This conformational change in Cmic leads to its structural change that it increases its recruitment to target genes and activation of those target genes. Phosphorylation at serine 62 also primes a second phosphorylation event at threonine 58, and this targets Cmic for degradation through the recruitment of the PP2A phosphatase B56 alpha, and then subsequent recognition by the FBW7E3 ubiquitin ligase. We've shown in cancer that the second half of the pathway gets turned down, leaving Cmic in a highly stable, transcriptionally active form that's highly serine 62 phosphorylated, as indicated here in this pancreatic ductal adenocarcinoma sample. KRAS is mutated in nearly all pancreatic ductal adenocarcinomas, and we've shown that it leads to phosphorylation of Cmic at serine 62, and this activates Cmic. We've observed this to occur early in pancreatic ductal carcinoma progression, and we see high phosphoserine 62 levels in pannins. Recently, a talented surgeon scientist and talented research assistant professor in my group looked at a large RNA-seq data set that we had generated, and they observed that activation of the hallmark MICV1 transcriptional pathway was enriched in patients who had undergone potentially curative resection, but rapidly recurred within 6 months. These so-called rapid recurs have high levels of Cmic transcriptional pathway activity compared to long-term recurs. We have generated a novel mouse model of Cmic and RAS-driven PDAC. This work was initiated by a talented postdoc in the lab, Amy Farrell, and has been carried on now by Isabel and Patrick. In this mouse model, we have knocked in the Cmic transgene downstream of a lock-stop lock into the constitutively active ROSA26 locus. We also have the lock-stop lock KRAS G12D, as well as a tamoxifen-inducible CRE that's expressed in the pancreas. When we turn on CRE with tamoxifen in the adult mouse, we then turn on mutant RAS and activate low deregulated levels of Cmic. In this mouse model, both the KRAS CRE and the Cmic CRE are not able to drive PDAC within 700 days. However, the combination of either one knock-in copy of Cmic or two knock-in copies of Cmic accelerates the development of PDAC and leads to 100% of the mice developing invasive metastatic PDAC. Amy originally noticed in this KMC mouse model that we observed spontaneous loss of the tumor suppressors P16 and SMAD4, as well as gaining nuclear P53. And these are all hallmarks of human pancreatic ductal adenocarcinoma. Similar to other people who observed that the standard KPC, KRAS P50, mutant P53 CRE model of PDAC does not lose SMAD4, we also see that it retains SMAD4, whereas our KMC tumors have lost it. Isabel has been making cell lines from tumors from this mouse model, and they have various morphologies in vitro. Importantly, orthotopic injection of these cells into the pancreas results in a highly stroma desmoplastic PDAC tumor that recapitulates the histology of the autophonous tumors from which it was derived. In addition, This mouse model is highly resistant to standard of care chemotherapies, including gemcitabine, as well as the combination chemotherapy FOLFIRINOX. Interestingly, several of the lines that have higher levels of stabilized Cmic protein are highly metastatic compared to lines that have lower Cmic. And in these lines with higher Cmic, we have reproducible metastases to the liver by 21 days post-injection into the pancreas that can be tracked by ultrasound. And here you can see an H&E histology of the resulting liver metastases. In terms of targeting Cmic, it has so far remained elusive for direct targeting. A talented postdoc in the lab, Brittany Allen Peterson, wrote a review on strategies for targeting Cmic, and she's currently an assistant professor at Purdue. Other strategies that people are taking include trying to block Cmic transcription, as well as trying to post-translationally block Cmic. And this includes drugs that would disrupt the Cmic-Max dimer, increase proteasomal degradation, inhibit the PIN1 proline isomerase, or activate the PP2A phosphatase. And this is one of the areas that we're focused on. PP2A is a very potent tumor suppressive phosphatase composed of a catalytic subunit, a structural subunit, and a variety of different B regulatory subunits. And we identified many years ago that the B regulatory subunit that targets PP2A to Cmic is B56 alpha. We're working with Gautam Narla, a professor at Michigan, to develop a small molecule activator of PP2A. This small molecule sits in the pocket between the catalytic and structural subunit and the B regulatory subunit, specifically the B56 alpha subunit, and nucleates the holoenzyme stabilization, thus targeting PP2A more specifically to Cmic. A prior talented postdoc in the lab, Megan Jolly, demonstrated that treatment with this small molecule activator decreases Cmic expression and can combine with chemotherapy. Brittany had also looked at the activity of this PP2 activator in vivo and demonstrated that treatment of mice with this inhibited early metaplasia that precedes pancreatic ductal adenocarcinoma, and showed that she had reduced acinar ductal metaplasia with treatment with the small molecule activator PP2A. Brittany, as well as Isabel, also showed that the standard KPC autophonous model, you could double the survival in these mice by treating with this small molecule PP2A activator. And a current postdoc in the lab, B.D. Sa, has been looking at phosphorylation of Cmic in treated tumors in vivo, and found that phospho-Cmic is dramatically reduced with treatment with the small molecule PP2A activator. I want to thank you for listening to my talk today. I tried to mention all the people involved in the work that I showed, and I want to thank my lab for all of their hard work, as well as our collaborators and my funding sources. Thank you very much.
Thank you, Dr. Sears. I hope you will be available later on to take questions in a virtual format. So the next talk is by Mandar Mazumdar. He's Assistant Professor of Genetics and Scientific Director for Center for Gastrointestinal Cancer at Simlo Cancer Center at Yale. The title of the talk is Endocrine Exocrine Reprogramming in Obesity-Driven Pancreatic Cancer.
I want to thank the organizers for the wonderful opportunity to speak about our work today, exploring the causal link between obesity and pancreatic cancer. My name is Mandar Mazumdar, and I'm a member of the Cancer Biology Institute and Department of Genetics at Yale University. I apologize that I cannot be with you in person today, and I look forward to your questions and comments in the live panel discussion. My lab is broadly interested in the genetic, environmental, and host factors that drive cancer progression, with a special focus on pancreatic ductal adenocarcinoma, or PDAC, a highly lethal cancer with a 5-year survival rate of approximately 10%. One such modifiable host factor is obesity. Numerous epidemiologic studies have shown that body mass index, a surrogate measure of obesity, is associated with a dose-dependent increase in the risk of developing PDAC. More recent studies have further shown that obese individuals are more likely to present with metastatic disease, leading to shortened survival compared to normal weight individuals, arguing that obesity can promote both the initiation and progression of PDAC. To define a causal role for obesity, many groups, including our own, have fed high-fat diets to KC mice an oncogenic KRAS-driven model that closely recapitulates the genetic and histologic features of human PDAC progression. Most prior studies have used lard-based high-fat diets and shown an enhancement in tumor development. Christian Ruiz, a postdoc in the lab, became interested in whether specific sources of fat influence high-fat diet-driven cancer progression, especially given the wide array of vegetable and animal fat sources that are consumed in modern human diets. Christian collaborated with Matthew Rodehofer at Yale and fed KC mice a large panel of isocaloric high-fat diets that differ solely in dietary fat source. He found that, indeed, the source of dietary fat had differential effects in tumor development, and he is currently studying how specific dietary fatty acids within these diets link to tumor genesis. To further confirm that obesity acts causally to promote PDAC progression, we crossed the KC model with the OB-OB genetic model, which exhibits rapid onset obesity to loss of the satiety hormone leptin. We observed a marked enhancement of tumor development in resultant KCO mice that corresponded with a dramatic reduction in median survival, comparable to that observed in mice harboring an additional mutation in the tumor suppressor gene P53. The leptin deficient model gave us a tractable tool to test whether reversion of obesity through weight loss could intersect tumor genesis. Lauren Lors, a research associate in the lab, used genetic leptin replacement or dietary caloric restriction to induce rapid weight loss, such that mouse weights returned to their lean non-leptin deficient siblings within approximately 3 weeks. She found that early but not late weight loss reduced tumor development, arguing that obesity plays a causal and reversible role in early PDAC progression. A number of mechanisms have been proposed to mediate the effects of obesity on tumor promotion, including alterations in tumor cell metabolism, immunity, and secreted factors derived from immune cells, adipocytes, or endocrine organs in the micro and macro environment. For the remainder of the talk, I will focus on the pancreatic islets of Langerhans, which represent a component of the pre-tumor microenvironment that is unique to PDAC. PDAC is commonly thought to arise from the exocrine pancreas comprised of digestive enzymes secreting acinar cells and duct cells. These exocrine cells are in close proximity to endocrine islet cells, and emerging data have shown both bidirectional blood flow between these compartments and multiple points of reciprocal interactions in diseases, including diabetes and pancreatitis. Therefore, we hypothesized that obesity could reprogram the endocrine pancreas as a local source of hormones, perhaps insulin, perhaps others, to drive exocrine tumor genesis. To address this hypothesis, Kathy Garcia, a graduate student in the lab, isolated primary murine islets for single-cell RNA sequencing from congenic wild-type mice and moderately obese high-fat diet-fed mice and severely obese OB-OB mice in collaboration with Dick Kibbe's lab at Yale. Following dimensionality reduction and visualization together with the Krishnaswamy lab at Yale, Kathy observed a trajectory of beta cell gene expression states that corresponded to increasing obesity. Along this trajectory, she surprisingly found a reduction in insulin expression that corresponded to a reciprocal increase in the expression of another peptide hormone, cholecystokinin or CCK. CCK is typically produced by neuroendocrine cells of the intestines and acts on pancreatic acinar cells to stimulate digestive protease production and release. It has been known for quite some time that exogenous administration of the CCK analog cerulean can promote KRAS-driven pancreatic tumor genesis. So we reasoned that obesity-adapted islets could similarly provide a local source of CCK that could stimulate exocrine tumor genesis. In support of this hypothesis, Jeremy J. Cox, an oncology fellow in the lab, found that exocrine acinar cells upregulated multiple digestive proteases in response to obesity, including PRSS1, alterations of which are associated with hereditary pancreatitis and pancreatic cancer. In subsequent experiments, we confirmed that islet beta cells in obese tumor-bearing mice also express CCK protein, and that expression is abrogated with weight loss. Similarly, we observed islet-specific expression in the majority of human PDAC tumors, and a positive correlation between body mass index and CCK mRNA expression in human donor islets. Together, these data support the conclusion that obesity induces islet CCK expression in both mice and humans. To test whether beta cell specific CCK expression is sufficient to promote pancreatic tumor genesis, Lauren crossed lean KC mice to a transgenic model in which CCK expression is driven by the mouse insulin promoter. She found that compound MKC mice exhibited both increased acinar cell proliferation and tumor formation, supporting islet CCK as a driver of exocrine tumor genesis. Importantly, islet CCK overexpression did not fully phenocopy the effects of obesity and tumor development, arguing for additional obesity-associated factors being involved in cancer pathogenesis. Therefore, it should not have come as a surprise when Jafar Singh, a former postgrad in the lab, treated obese KCO mice with the CCK receptor antagonist proglumide and did not observe a reduction in tumor development. This could certainly have been due to the inability of the competitive inhibitor to overcome the presumably high local concentrations of islet-derived CCK. And we are currently testing this hypothesis using conditional genetic knockout methods. Alternatively, additional islet-derived factors could play a pathogenic role. In support of this latter hypothesis, we found that islet prohormone gene IAPP displayed concordant upregulation with increasing obesity in both non-tumor bearing and tumor bearing mice. Based on these data, Lauren used an available mouse transgenic model allowing islet-specific overexpression of human IAPP and observed a marked increase in KRAS-driven tumor development. Given that multiple aberrantly expressed hormones could play pathogenic roles in obesity-driven PDAC progression, we sought to understand the upstream mechanisms that led to islet reprogramming. CCK has been shown to be a beta cell survival factor in the setting of toxins or stress. Indeed, we observed an upregulation of genes associated with protein translation, secretion, and proteotoxic stress in beta cells from obese OB mice that corresponded with beta cell dysfunction shown by reduced in vitro glucose-stimulated insulin secretion. We reasoned that this stress response could be due to increased insulin demand in the context of persistent obesity-associated hyperglycemia. So we treated KCO mice with the anti-diabetic SGLT2 inhibitor dapagliflozin, which lowers blood sugar by blocking glucose reabsorption in the kidney. Indeed, DAPA markedly reduced average blood glucose in the KCO model, which resulted in the improvement of beta cell function measured by higher circulating levels of insulin and proinsulin in the majority of treated mice. Surprisingly, analyses of tumor burden in these mice revealed a negative correlation with insulin production. Mice exhibiting high levels of tumor burden had low islet insulin protein and high levels of CCK. In contrast, mice with low tumor burden had high islet insulin and low CCK expression. Together, these data support a correlation between CCK rather than insulin in tumor development, arguing that endocrine exocrine signaling beyond insulin may be pathogenic in obesity-driven PDAC development. During the course of the talk, I hope I've convinced you that obesity plays a causal and reversible role in early PDAC progression, that obesity reprograms the pre-tumor microenvironment, including the endocrine pancreas, to alter signaling to the exocrine pancreas, and that these pathogenic alterations go beyond insulin, and that islet modulation can enhance or subvert pancreatic ductal tumorigenesis. Therefore, I would argue that targeting the endocrine pancreas could be leveraged to intercept exocrine tumorigenesis. I want to thank the many lab members who contribute to this work who I highlighted along the way. We're indebted to collaborators at Yale and beyond and to multiple sources of critical funding for this work. Finally, we're seeking postdocs to join our diverse, collaborative, and fun-loving team. So please reach out. Thanks, and I'll take it.
The session is open for question and answer. Can we have the online speakers also? Dr. Ramki.
Hello?
For Pablo. Autophagy has been linked to chemo resistance. If you have increased autophagy, many other tumor models they have shown. In your case, you are knocking out ATG5 heterozygous. So in that model system, are they more sensitive to chemo?
No, they are not sensitive. They are resistant to chemotherapy. We have tried this. I couldn't show the data because of the time frame, but we treated them with gemcitabine. They were more resistant.
Oh, thank you.
Morning, everyone. My question is for Dr. Sears. So great work. What I wanted to ask you is if you did any genome-wide association studies to look at the transcriptional profile of the PP2A inhibitor that you used?
Yeah, hi, can you hear me?
Yes.
Great, thank you for the question. So we keep meaning to do the genomics on the PP2A activator, and we have not looked at transcriptional changes that arise from it its treatments. So either in vitro or in vivo. The PP2A activator that we're working with is selective to the PP2A B56 alpha holoenzyme. And so a lot of the activity will be due to its ability to block Cmic phosphorylation, but it can also affect other, I think that can target beta-catenin also. So there's going to be other transcriptional effects, and we have not tested that yet, and we need to. Thank you for the question.
The mice data look pretty promising. Are you thinking about coupling it with immunotherapy and to see if the tail could go up and the survival could be increased further?
Yes, we have actually done that in our, we also have a triple negative mouse model that's kind of similarly driven by low deregulated Cmic and loss of P10 in that case. And we've done it for that model, and we're just getting started doing it with the PDAC model. And it actually does combine nicely, and we've really were able to stop tumor growth much more robustly and increase survival. So it combined well with anti-PD-L1.
My last question, real quick, was for Dr. Mazumdar. Great talk, sir. In one of your introduction slides, you gave us a nice whole slew of the oils and fats that were utilized essentially, but there was no association with the canola oil as far as the tumor burden was concerned. Can you comment on that real quick?
Yeah, so we didn't actually test canola oil within our diet panel. It turns out that canola oil formulations can vary quite significantly from one canola oil to another. So since we couldn't sort of generate a standardized oil diet that we could use for canola oil, we did not include it in the panel, despite the fact that it's frequently consumed. Again, we wanted to control the fats very carefully.
Hello. Yi Yang from UNC Chapel Hill. I have a short question for Dr. Sear. Wonderful talk and interesting observations of Cmic. I'm curious, when you observe high activity Cmic in the PDAC recurrent patient, are those Cmic, high activity of Cmic due to amplification of the Cmic or transcriptional activation of Cmic or something like that? Because the reason I'm asking this is because at least in preclinical model, when you treat with PDAC in lung or in lung cancer KPC model with all kinds of signaling pathway inhibitors, and when the tumor gain resistance, one of the reasons is people observe is the Cmic amplification. So I'm wondering whether there's a correlation between two.
Yeah, that's a good question. So we have the cohort we're looking at in which we have transcriptomics and exome sequencing is 300 PDAC cases, and out of those, I think there were 35 of these rapid recurs. I want to say, I don't remember the exact number. There was no statistically significant increase in Cmic gene amplification, but there was a significant increase in the Cmic hallmark V1 transcriptional signature and also in phospho-Cmic staining. So I think that it can be driven by gene amplification, but also by post-translational stabilization of Cmic and activation of it Cmic post-translationally to also give you that high Cmic transcriptional signal sequence, the transcriptome. So I think you can get at it either way, but we did not see an enrichment in amplification.
Thank you.
So my question is for Dr. Shields. So there is you use this PP2A activator. So it's targeting the catalytic subunit of PP2A.
It binds in the pocket between the catalytic and structural and B56 alpha. So it binds in the pocket of the trimeric holoenzyme.
but only with the B56 alpha subunit. So PP2A has a conserved catalytic and structural subunits, and then a number of different B subunits that target it to different substrates. And this small molecule doesn't bind in the catalytic domain. It doesn't block, it actually increases the catalytic activity, not simply by nucleating the holoenzyme formation.
So you mentioned that the phosphorylation sites, one is serine 62 and the threonine 58. So have you ever looked at the threonine 58 phosphorylation controlling this pathway?
Yeah, we have, and you know, it's not actually clear why PP2A is not, at least the PP2A B56 alpha holoenzyme is not dephosphorylating threonine 58. So it dephosphorylates serine 62, but we don't see it hitting threonine 58. But there is another report arguing that there's a, initially it was thought to be a different protein complex, but then it was shown to include PP2A. And I think that was a B55 alpha subunit that could then dephosphorylate threonine 58. So it was a different one of the PP2A holoenzymes. And it's actually, although PP2A is primarily a tumor suppressive phosphatase, and you know, its suppression is almost universal in human cancers. There are trimeric holoenzymes of PP2A that are more tumor promoting, and that's one of them. With the B55 subunit.
Hi, this is Koelina from UNMC. I have a question for Dr. Mazumdar. Wonderful talk. I actually wanted to ask, in your CCK transgenic model, did you guys see any change in the microenvironment, as in fibrosis, or you know, the peripancreatic fat, or in the visceral fat? That, I mean, I wanted to ask that for this, both for CCK transgenic model as well as for your obesity model.
Yeah, so in our obesity model, we did see by bulk RNA sequencing, as well as by histologic analysis, increased evidence for increased inflammation and fibrosis. In the islet CCK transgenic model, in the absence of tumors, interestingly, it does not cause sort of local pancreatic inflammation in the short term. Eventually, the mice do at about a year of age and beyond. But within the time frame we were analyzing tumors, we didn't see any evidence of inflammation. And the group that generated the mice as well has looked at this, and they have not seen any inflammation induced by the islet transgenic within sort of the first year of the mouse's life, suggesting the possibility that islet CCK is functioning independent of its pro-inflammatory function to promote tumorigenesis in the non-obese KC model, though we can't exclude a possibility for inflammation also playing a role in the context of the KRAS mutation.
Thank you. And so do you see in either of these models any mobilization of adipocytes or adipose mesenchymal stem cells surrounding these tumor tissues, or towards the periphery, anything mobilizing from the peripancreatic fat area?
It's a great question. We haven't looked carefully at it. It's something that we want to, and actually I have a MD PhD student joining the lab who's very interested in adipocyte biology. So he's already started to do some of those isolations, and so we'll see what he finds in these models.
Thank you so much.
I have a question for Dr. Mazumdar.
Sure.
That was a beautiful talk. I was wondering, you know, in the non-obese setting, so just in the normal diet mice, do you also see, I mean, you know, when we look at the histology of PDAC, there's islet hyperplasia, as well as the islets kind of get scattered and broken up, and so there's a lot of crosstalk between the islets, and even non in an obese setting. Do you think that all the pathways you're studying also play a role there, or only in an obese setting?
So we've examined this in sort of in tumors that have evolved in KC and KPC mice that are non-obese over time. And we see the same things in sort of alteration of islet architecture, invasion of the islets. Interestingly, we don't see the same effects on CCK expression, for example, by immunohistochemistry in the KPC or KC mice, only in the KCO models and in some of our high-fat diet models. So I do think there is a different effect that's going on there in terms of islet dysfunction. Now, it's well known that pancreatic cancer in a subset of patients can induce new onset diabetes. And so there's likely crosstalk in human populations that leads to islet dysfunction. But it's an area of active investigation, at least in our non-obese animals, in which we get multifocal tumorigenesis. While we see alterations in islet architecture, we don't see the same effects on islet hormone expression, for example.
So, you see a lot of changes in cytokines when the autophagy is inhibited. So what's the mechanism of connection of these autophagosomes to cytokine production?
That's a very important question. I mean, this is something that we are currently trying to elaborate, how the heterozygous deletion of ATG5 affects the secretion. So this is something that I cannot conclusively answer right now, but this is something that we are trying to look in.
And what's your hypothesis, or like why? So the hydrochloroquine works so nicely in the animal models, but didn't actually had a opposite effect in the clinical trial. What's the the current thought process on that?
Well, this is something that we have experienced in many fields, right?
Sure.
So,
Yes,
but,
absolutely. Yeah.
I think when it comes to autophagy, this is a broad view right now, what we are trying to have on autophagy, but it's much more complicated than looking on autophagy flags and inhibiting hydrochloroquine. I think we have to look at the components of the autophagy machinery as well, and to see how they are linked to the effect of autophagy inhibition. I think that's much more complicated than we initially thought. This is something that we can say right now.
Dr. Ramki.
I have one general question for all the three speakers. It is now well accepted in the tumor biology that there is a metabolic symbiosis going on within the tumor microenvironment, where some of the metabolites from tumor is converted by the stroma and then feeding back to the tumor. So whether it is a Cmic or OB or CCK8 or top ATG5, if you make the change in the stroma, does it affect tumor growth? Or is there, can you, from your model system, can you say anything about this metabolic symbiosis between tumor and stroma?
Hannah, you want to take?
Go ahead.
Go ahead.
Well, just there's beautiful work that Mara Sherman has done showing the role of fibroblasts in the stroma in terms of the release of fatty acids and feeding of the tumor cells. We have been doing some studies looking at the PIN1 proline isomerase specifically in the stroma, and if we knock that down, and then if we have that knocked out in the host, and we put in KPC cells, they don't form tumors robustly. They're highly diminished in size. And so I think there for sure is a role. And what we've shown is that affects the ability of the fibroblasts to take on a MyCAF phenotype. And then they don't release as much HGF, which is the ligand for the MET receptor and such. So I think there is, I mean, of course, there's strong crosstalk. And if we can eliminate signals from the stroma, I think it can have a robust effect on the tumor cells.
I have one question for Manda. I would like to challenge your concept, because currently it is generally accepted that KRAS mutation occurs not in the acinar cells, at least in the human setting. So how can you bring both concepts together? CCK acts on acinar cells, but KRAS is usually not mutated in acinar cells. How is this conundrum? How can this conundrum be solved? So what is your thoughts on that?
I think it still remains controversial what the cell of origin is in both human
Yes.
and mouse cancer. Though in mouse cancer, I think most of the models probably recapitulate an acidotic ductal metaplasia that then leads to ductal tumorigenesis. I think that there are probably multiple hormones that are at play. I showed you some evidence of another hormone, IAPP, that could play a role, the receptors of which are not well established in the exocrine pancreas, but there are other aberrations in the endocrine pancreas as well. So I think it is likely that there could be multiple cells of origin, and there could be multiple tumor promoting stimulants that are coming from the endocrine compartment that can act on individual cells within the exocrine compartment to drive the initial events in tumor formation.
Well, this was great discussion and great session. But
Can I ask one more question to Dr. Agul? That was a beautiful talk. I was wondering, have you noticed any connection between your work with ATG5 and cachexia, or just even in the human population? Is there a strong connection between the degree of autophagy in a patient's tumor and their degree of their cachexia?
Well, this is a very important question, but trying to answer this question, you have to have patients that will give you some more samples than pancreatic cancer itself. So I think if you want to analyze autophagy in the setting of cachexia, you have to have access to muscles, to fat. And so this is something that we do not have. So it will be very difficult to analyze this in the human setting. When it comes to the mouse models, we are currently trying to analyze the link of autophagy activation as well as inactivation and the impact on an adipose tissue, and as I showed with one slide, on other organs like in the liver. So this is something that we are currently trying to elaborate in more detail, but that's a very important issue, I think, when it comes to cachexia of patients, to understand that in more detail.
All right, thank you all for joining, and we will come back after a small break. Thank you.
Thank you.
Thanks.